Our second speaker is Waraporn Taweesin from Khon Kaen in Thailand. For those of you who've been, it's a fabulous place. Who's going to tell us about transcriptomics between in flute-related clanjo and prevention. Thank you very much indeed.
Yep. Hi everyone, good afternoon, and thank you for having me. I think that all of you might have heard about this species that I'm going to talk today. I would truly represent about about my work. Since the late 19th century, the OV or Opisthorchis viverrini was found as a sea-level fluke, and it's the major risk factor for CCA development. And especially in Thailand, from the eating of raw or uncooked fish that infected with the OV is a major risk factor for this disease or liver fluke infection. So, especially in the Thailand northeastern region, we found that this is found with high incidence of OV infection, not only in Thailand, but other countries in Southeast Asia as well. For example, Vietnam, Laos, and Cambodia also found this species. Under the cholangiocarcinoma screening and care program, or CASCAP in Thailand, that work with the Khon Kaen University, from the Isan people or northeastern people, population around 24 million, we found that this group were detected as the positive OV infection with around 9% of showing here. And for the raw number is around 100,000 people, which is still low for this number. So the matter question that comes in my mind is that if we can increase this number from the efficient diagnosis or prevention, we can improve the, we can detect the early stage of CCA that the people is going to turn to. So, and how we can access that? One of the things is that the identified candidate biomarker or the finding of drug or vaccine targets is one of the possible things. And before we access that, we have to understand the parasite biology of this worm within the membrane host. For the information that I would like to represent is that from the previous study showing us that the transcriptomics changes of this stage around 2 weeks and 1 month show the difference and some similarity between the stage. And for this stage is quite newly for us. We add more to understand what is happening in the worm during they got the infection by parasite, and what is the biological process in the parasite in parasite that respond to the host. So the method that we select for is that transcriptomics, or the study of mRNA molecule to understand what is happening, especially biological processes. And after we collected the metasaccharide from fish, we infect to the hamster, which is using as the animal models in this study. And after infection at different time point, we extract the RNA for the sequencing. And also obtaining the raw data, data analysis, especially we focus on differentially expressed gene for for understand the biological process with goal enrichment or goal ontology enrichment. So what we found as our expectation, there is something that shows us the juvenile have the different gene expression pattern between the adult stages, both up to here, whereas the adult stages show some similarity from the gene expression data. And we look further about the state-specific genes, and we found that when we compare the gene profile between the adult and juvenile, we found around thousands of genes were down-regulated. At the same time, around over 500 and 900 were upregulated. So, but not different too much between the acute and chronic infection. just 5 genes that show us. And we look further for the function of the gene from Go ontology enrichment, and we found that the biological process that enrich in the juvenile stage or 14 days post-infection. We found the gene involved in proteolysis here, around 25 genes, and multiple genes also involved in neural signaling process or signaling cascade, and also enrich the ATP synthesis or energy metabolism. And this evidence indicated that the worm at the juvenile stage might have the faster development during they are the juvenile inside the bile duct of the host. And furthermore, what's happened in the adult, both of stages, we found the gene that upregulated were related in the reproduction process. For example, overprotein, sperm associated antigen 6, tyrosinase, and trematode eggshell synthesis. And furthermore, we also found a gene involved in proteolysis as well. For example, leguminous peptidase that also involved in the host immune system. And furthermore, the plasminogen or the gene that involved in the fibrolytic degradation were found as related in the parasite immune system. So this is, we assume that this evidence at the adult stages, both of adult stages of the worm can promote themselves to stay in the host longer and longer. And with the same data set, we also analyze the gene with stable expression or housekeeping gene to find the best one for being act as the candidate biomarker for Opisthorchis. And around 600 non-human ortholog. In total, around 2,000 genes were assigned as the non-human ortholog. I just show you about top 15 genes here that select from those of highest expression level, because we would like to improve about the better sensitivity for diagnosis. So this is the one that I would like to recommend. For example, tubulin alpha chain, some gene that didn't annotate at the exact name also involved in this study. So to conclude my talk, in this study we didn't understand, we not only understand the parasite biology, but we could provide the key biomarker or drug and vaccine target for improve the diagnosis, or also I hope that it might be useful for prevention for CCA as well. And finally, thank you so much for all teamwork, all relevant people, my laboratory, and also relevant organization. And thank you so much for this conference that gave us the opportunity to share this work. Thank you so much.